How does recombinant DNA create better immune vaccines? Have you ever wondered how scientists create vaccines that help our bodies fight off diseases? One fascinating method is through recombinant DNA technology. This approach allows for the design of vaccines that can effectively train our immune systems. Let's break down how this works and why it matters. Recombinant DNA technology focuses on creating specific proteins called antigens. These proteins are crucial because they trigger an immune response. Instead of using whole viruses or bacteria, which can sometimes cause illness, this method uses only the genetic instructions needed to produce these antigens. When these instructions enter our bodies, our cells can make the proteins themselves. This process mimics a natural infection. Our immune systems recognize these proteins as foreign invaders. This recognition prompts our bodies to mount a defense. The result is a targeted response that prepares our immune systems for future encounters with actual pathogens. One of the key benefits of this technology is efficient antigen presentation. The proteins made by our cells are displayed on their surfaces. This display uses special molecules that alert immune cells, particularly T cells. These T cells play a vital role in identifying and responding to specific threats. Recombinant DNA vaccines also activate both arms of our immune system. They stimulate B cells, which produce antibodies, and T cells, which can kill infected cells. This dual activation is essential because it helps our bodies build a robust defense against infections. Additionally, the DNA used in these vaccines can engage our innate immune system. It contains patterns that sensors in our immune system recognize. This recognition triggers the releases of signaling molecules that enhance the immune response, making the vaccine even more effective. Safety is another important aspect. Since these vaccines only use selected antigen genes, they avoid the risks associated with whole pathogens. This means there is a lower chance of causing infections or adverse reactions. The antigens produced closely resemble the natural proteins of the pathogen, ensuring our immune systems learn to recognize real threats accurately. Flexibility is a significant advantage of recombinant DNA technology. Scientists can quickly design and produce vaccines by inserting genes for new or mutated antigens. This speed is crucial when responding to emerging infectious agents or variants. When we talk about subunit vaccines, which include only parts of a pathogen, recombinant DNA technology shines. It allows our immune systems to focus on the most relevant components without unnecessary exposure to harmful parts of the pathogen. The expressed antigens undergo natural processing, helping present them in their native form to immune cells. In summary, recombinant DNA technology creates better immune vaccines by using our body's own systems to produce authentic proteins. This method engages both innate and adaptive immunity, providing a safer and more targeted vaccination strategy. By harnessing this technology, we can improve our defenses against diseases and enhance public health outcomes.